So today, I'm talking about, I call it actually brain alive. I was thinking of a term to use because I don't know, it just sort of came to me. But a little background, I actually did research. I wanted to go into neurosurgery many years ago in training in Canada. And so I have a passion in my heart for neurological problems and issues. I also come from Montreal, where there's a Montreal Neurological Institute, where a famous surgeon mapped the brain out. So I sort of have this background history of interest in neurology, neuroanatomy, and neurophysiology. And we're going to talk about brain health today. We're going to talk about brain and disease and brain and healing and what we can do with Cerule products and how they work together. I've got a lot of data I need to get through, so I'm going to start right away. I'm not going to move around as much, so that I've heard some good feedback about me sitting still. I'm just used to talking in front of live audiences, so I kind of one of those guys that move around. But anyway, I want to focus on the human brain. Our brain only weighs 3.3 pounds. It will triple in size in the first year of life, yet the average adult human brain has about 100 billion cells. The cells are connected by synapses, and each cell could connect to tens of thousands of other cells. So if you do the math, the possible permutation combinations of connections in the human brain is somewhere like 100 billion to the power of 10,000, which is 1 times 10 to the power of 110,000. To try and put that into perspective, there are about the same number of stars in our galaxy as brain cells in the human brain. Yet the possible connections, as I mentioned, make the brain 10,000 times more complex than our galaxy's stars. So let that sink in. So if you think sometimes you're carrying the world on your shoulders, you're actually carrying the galaxy on your shoulders. According to the Dent Neurological Institute, exercise, we're going to talk about different things about health, about and our brain. Exercise is just as good for your brain as it is for your body. Aerobic exercises raises your heart rate, increases blood flow to your brain. As your increased breathing pumps more oxygen into your bloodstream, more oxygen is delivered to your brain. This leads to neurogenesis. Genesis means growth, or the production of neurons in certain parts of your brain that control memory and thinking. Neurogenesis increases brain volume, and this cognitive, that means thinking, reserve is believed to help buffer against the effects of dementia. So one of the things I want to dispel today is that you think, you're getting older, but and you can't do as much mentally, or I'm kind of giving up or whatever, and I can't grow new nerve cells. It's sort of, we were told many years ago and decades ago that the brain is sort of static, and what you get when you're older, that's it. That's completely wrong. So I'm here to explain that to you. And this is good, because there's lots of good things we can do for our health, including, of course, the all important STEM Enhanced Ultra. And I'm going to talk about the products obviously in a little bit. It's been noted that exercise promotes production of neurotrophins, leading to greater brain plasticity and therefore better memory and learning. In addition, neurotrophins, these chemicals, exercise also increases and allows an increase in the neurotransmitters, specifically serotonin and norepinephrine, which boosts information processing and mood. So I mentioned last time that I would talk about something called neuroplasticity. It's something that I think is really cool, because I do a lot of extreme sort of physical activity, CrossFit, mountain biking, and ice hockey, and so on. And I've noticed that if you push yourself and you kind of hand-eye coordination and spatial movement, that you can actually get better. It's not just your coordination, but actually I think your memory gets better, and we're going to talk about that. So a person called Eleanor McGuire documented changes in the hippocampus structure of the brain associated with acquiring the knowledge of London's layout by local taxi drivers. So she asked these taxi drivers to run different routes and try to remember. And she found that there was a redistribution of gray matter was indicated in these drivers compared to control. So just by driving around town and challenging them to drive was enough to trigger memory and triggering certain cells in the gray matter. A fellow named Michael Merzek is a neuroscientist who has been one of the pioneers of neuroplasticity for 3 decades. He says the most ambitious claims of the field that brain exercises can be made useful as drugs to treat diseases as severe as schizophrenia, that plasticity exists from cradle to grave, and that radical improvements in cognitive functioning, how we think, learn, perceive, and remember, are possible even in the elderly. Merzenk's work was affected by a crucial discovery made by Hubel and Wiesel in their works with kittens. The experiment involving, it sounds a little crass, but anyway, sorry, bear with me, involves sewing one eye shut and recording the cortical brain mapping. Hubel and Wiesel saw that the portion of the kitten's brain associated with the shut eye was not idle as expected. Instead, it processed visual information from the open eye. It was as though the brain didn't want to waste any cortical real estate and had found a way to rewire itself. So this is important for everybody. So whether you're swimming in the ocean, like some of us are lucky to do, or anything active, that's very important. Merzenk's first encounter with adult plasticity came when he was engaged in a postdoctoral study with Clinton Woosley. The experiment was based on observation of what occurred in the brain when one peripheral nerve was cut and subsequently regenerated. The two scientists micromapped the hand maps of monkey brains before and after cutting a peripheral nerve and sewing the ends together. Afterwards, the hand map in the brain that they expected to be jumbled was nearly normal. This was a substantial breakthrough. Merzenk asserted if the brain map could normalize its structure in response to abnormal input, the prevailing view was that we are born with a hardwired system had to be wrong. The brain had to be plastic. Merzenk received the 2016 Cavalli Prize in Neuroscience for this discovery. So, neurobiology, neuroplasticity as the ability to make adaptive changes related to the structure and function of the nervous system. And this concept actually was started like 100 years ago. There were thoughts about this. It's nothing new, but it was pushed aside and thought to be crazy. Correspondingly, two types of neuroplasticity are often discussed: structural neuroplasticity and functional neuroplasticity. So one of the things, so we're talking about neuroplasticity, we're going to talk about Parkinson's, we're going to talk about multiple sclerosis and ischemic brain injury, and how does Cerule products interact with all of these problems and in health as well. So structural neuroplasticity is often understood as the brain's ability to change its neuronal connections. New neurons are constantly produced and integrated into the central nervous system throughout the lifespan based on this type of neuroplasticity. Researchers nowadays use multiple cross-sectional imaging like MRI scans, CT scans, to study the various alterations of the human brain. This type of neuroplasticity often studies the effects of various internal or external stimuli on the brain's anatomical organization, how we're wired. The changes of gray matter proportionate to the synaptic, that's a connecting, strength in the brain are considered examples of this. Functional neuroplasticity refers to the brain's ability to alter and adapt the functional properties of neurons. The changes can occur in response to previous activity, to acquire memory, or in response to malfunction or damage of neurons to compensate for a pathological event. That means a brain injury or a disease. In the latter case, the functions from one part of the brain transfer to another part of the brain based on the demand to produce recovery. So just imagine the brain is working with other parts of the brain to auto-correct itself. I mean, it's mind-blowing. Anyway, application examples. The adult brain is not entirely hardwired with fixed neuronal circuits. There are many instances of cortical and subcortical, so the cortex, the top part, subcorticals below, rewiring of neuronal circuits in response to training as well as in response to injury. There is evidence that neurogenesis, birth of brain cells, occurs in the adult mammalian brain, and such changes can persist well into old age. And there's multiple references for this data, which I can share. The evidence for neurogenesis is mainly restricted to the hippocampus and olfactory bulb, but current research shows that other parts of the brain, including the cerebellum, which is the hind part, which is responsible mainly for balance. Okay, treatment of brain damage. A surprising consequence of neuroplasticity is that the brain activity associated with a given function can be transferred to a different location. This can result from normal experience and also occurs in the process of recovery from brain injury. Neuroplasticity is the fundamental issue that supports a scientific basis for treatment of acquired brain injury. Thus, neuroplasticity is gaining popularity as a theory that explains improvements in functional outcomes with, for instance, physical therapy when somebody has a stroke. So I think that's sort of our way of introduction and why I think it's really important to talk about neuroplasticity. And it's a word that I think we should sort of start banding about, start talking about, getting people to understand it, because that will make people realize that they can get better, they can improve. Don't just sit there, people have a stroke and they go see physiotherapy. It's like, do more, push yourself. You need to push yourself. Pushing yourself basically means triggering other neurons to be active. We want them to be active, don't sit around. So now that we know how amazing the brain is and what it's capable of doing, let's see what Cerule products can do to support and promote this brain development in plasticity and in recovery from disease such as multiple sclerosis, Parkinson's disease, mood disorders like depression and ischemia. So there's actually 10 papers. I know it sounds a lot, but we'll summarize them fairly quickly. AFA and the nervous system is the first name of the first paper: attention deficit disorder, depression, and mood elevation. AFA contains a significant concentration of phenylethylamine, PEA. This is a brain chemical known to increase our concentration, elevate our mood, and alleviate depression, coined the molecule of love. Dopamine, one of the main neurotransmitters in the brain, and there are many synapses based on dopamine. It turns out that these dopaminergic receptors, where the nerves meet, that's a receptor, are stimulated by this PEA stuff. Thus, PEA can improve our drive, pleasure, and generally make us feel good. This means we have less chance of becoming depressed. Major depression leads to a lack of motivation and drive, of course, not just finding things fun anymore, and it's called anhedonistic. We don't want to be that. Urine metabolites in patients who had PEA were lower in depressed patients than non-depressed patients, again, pointing to the importance of PEA. That's again phenylethylamine, which is in AFA, which is in our products. Stem Enhance Ultra has AFA, which contains PEA and thus triggers dopamine in the brain. Will make us happier, have more drive, and enjoy life more. Who doesn't want that? So that, I thought that was a really cool, interesting paper connecting AFA and this chemical in our brain that's really important that works with dopamine, the molecule of love. The second paper is cognitive enhancement and immune modulating properties of an antioxidant-rich microalgae, AFA NIN+. That was Gita Jensen's paper. And AFA IN+ contains PEA. Other algae do not. So selective evolutionary process somehow has led to PEA in AFA. That's kind of interesting. They also state that the phycocyanin is an antioxidant, of course we know that, and selective COX-2 inhibitor, which is an anti-inflammatory, as I think you all know by now. In addition, this study looked at reaction times and ability to multitask. Both of these improved after AFA ingestion in the subject versus the placebo controls. These improvements were seen within 2 to 10 minutes after consumption and continued for 1 hour after. So I thought that was also very interesting, the fact that it happened fairly quickly and was sustained. Third paper, phycocyanin, a bilirubin and antioxidant, anti-inflammatory and neuroprotective effects. Author Rome et al., Current Protein and Peptide Science, 2003. Phycocyanin is a constituent of spirulina, as you probably know. They summarize 12 different studies at lab animals, rats, and mice from different authors. So these are 12 studies summarized. The major theme was the antioxidant effects of the phycocyanin. Phycocyanin was a potent free radical scavenger. That means it reduces oxidative stress. We will look later at why reducing oxidative stress is important in brain injury and disease. Again, it was also shown to be a COX-2 inhibitor or anti-inflammatory agent. And of course, anti-inflammatory and oxidative stress reducer work well hand in hand. As a neuroprotector, in rat cerebellar brain cells, that's the one at the back of the brain, in culture were treated with a known agent to cause cell death, and phycocyanin reduced that rate and protected brain cells. So they said there was some protective effect that phycocyanin had on cells that were being intentionally injured in a dish. The fourth paper is called Protective Effects of Phycocyanin Against Canic Acid-Induced Neuronal Damage in Rat Hippocampus, author Rimbaud et al., Neuroscience Letters, 1999. Canic acid is a known agent to cause convulsions in rats and destruction of hippocampal cells. Thus, it is used to study epilepsy in humans by implication. But let me just, I mentioned hippocampus a few times here. Let me just quickly kind of show you a little brain map. So this is the brain kind of through here. This is the Sylvian fissure here. This is the parietal lobe, parietal lobe. This is the frontal lobe. So this is where the cortex is with all the connections that look sort of like, like a brain. And then this is where actually the hippocampus lives inside here. It's a small little structure. It's tucked away here, and there's one on each side. So when we talk about hippocampus, I'll just write hippo. That's what we're talking about, this little structure in there. But it's very, very critical to a lot of things that we're going to talk about, like memory. Okay. So the seizure activity was reduced in canic acid treated rats who received phycocyanin. There was a dose, this was dose dependent, which means the more they gave, the better they got, suggesting the more you take PC, the better phycocyanin. Also, when the hippocampal cells were studied, less neuronal damage was seen in the phycocyanin treated group. So that was very fascinating. So again, they had a model of damage to cells that were in the hippocampus, and phycocyanin reduced that damage. So it's the area, as we mentioned, the hippocampus is part of the limbic system, and it's responsible for short and long-term memory, as well as spatial memory that enables navigation, like those London taxi drivers I was talking about, and finding your way back home after you had a few drinks. In Alzheimer's disease and dementia, the hippocampus is the first region of the brain to suffer damage, leading to early symptoms of short-term memory loss and disorientation. As it gets worse, the patients will have trouble with anterograde amnesia, the inability to form and retain new memories. But neuronal plasticity helps form new memories here. Studies show that with age, the size of the hippocampus actually shrinks, which leads to poor memory formulation. This is also seen with post-traumatic stress disorder, like for instance, in Vietnam vets. Their hippocampus has shrunk by 20%. The good news, in 2011, a randomized control study showed that aerobic exercise increased the size of the hippocampus in adults age 55 to 80. Those are the ones that they studied. So actually, exercise increased the size of the hippocampus. So, which obviously means there's more nerve growth. And these are patients that were 55 to 80, not 25 years old. Good news, everyone. Paper 5. Spirulina prevents memory dysfunction, reduces oxidative stress damage, and augments antioxidant activity in senescence, aging, accelerated mice. This is authors Huang, Journal of Nutritional Science and Vitaminology, 2011. These mice mimic Alzheimer's disease patients. They have memory loss and spatial disorientation. Alzheimer's patients have deposits of this stuff called amyloid beta protein deposits in their hippocampus and other areas of the brain. Mice were randomized into 3 groups: control group with no spirulina, 2 treatment arms, one with low dose spirulina and one with a higher dose of spirulina. Mice that received spirulina had less amyloid deposits, those damaging things I was telling you about, in their brains, and had better memory and spatial orientation than the control did. And again, the higher the dose, the better. So again, when you're talking about how much to take, you need to increase your doses of these products to see where you need to be in terms of your maximum dosing to get maximum benefit. Parkinson's disease. Okay, so now everybody's heard of Michael J. Fox. He has obviously a Parkinson's Foundation, which is actually pretty cool. If you want to check out his website, there's lots of interesting information. Parkinson's disease is the damage of cells in the substantia nigra in the midbrain. So hang on a sec. So the midbrain is down here. So all the information from the brain goes down through the midbrain inside here. Well, it's inside there, not outside. I mean, not near the hippocampus, more underneath the brain. And then it goes down to the spinal cord. So if you look at the base of the brain from below, the spinal cord will be somewhere here. The hippocampi will be here. And all the information, of course, goes down through the spinal cord, this way. So this is the midbrain and through here, and the medulla oblongata. So the substantia nigra is in the midbrain, and this leads to a deficit in dopamine. The cause is not known, but there's a buildup of proteins called Lewy bodies in the neurons. It starts usually over age 60. The typical symptoms include tremors, weakness, stiffness, rigidity, slowness of movement, and difficulty walking. Again, we're talking about Parkinson's disease. Sometimes cognitive and behavioral problems can occur. Dementia occurs at later stages. Depression and anxiety can also occur. In 2015, it was estimated that 6.2 million people had the disease globally, which resulted in 117,400 deaths. The male to female ratio is 3:2, so more men than women. The average life expectancy following diagnosis varies from 7 to 15 years. But again, I think that, this is data from 2015, so it's 5 years old. But I think also that it doesn't take into consideration what we're talking about. If people understand their health more, eat better, exercise more, take Cerule products, I think that life expectancy can increase. There's no cure or treatment yet. We can only improve their symptoms with L-DOPA. But again, if you're bringing blood supply, increasing neural growth and neuroplasticity, I think you can extend that. There is, of course, surgery to install microneedles for deep brain stimulation has been used when the disease is severe. And prevention, exercise again in middle life may reduce the risk of Parkinson's. Caffeine can also help prevent it. So that's interesting. There's suggestions that non-steroidal anti-inflammatories can also help. A 2010 study suggested the use of NSAIDs resulted in a 15% reduction in incidence of the disease. Mortality risk factors, onset at older age, swallowing disorders, and dementia. New studies with gene therapy involving growth factors and enzymes such as glutamic acid decarboxylase have shown some promise. Trials are ongoing. So as we can see, the medication for Parkinson's is limited to symptom control and side effects. If we can do anything to help improve this horrible disease, we should do it. And that's what we're talking about today. Paper 6, supplementation of spirulina improves lifespan and locomotor activity in paraquat-sensitive, sounds weird, DJ1B flies in Parkinson's disease model in Drosophila melanogaster, which is a type of fly. Been studied for decades. This is Kumar et al., Journal of Diet Supplementation, 2017. This fly model of Parkinson's showed that spirulina improved the motor activity of the flies and their lifespan. They found a decrease in superoxide dismutase in spirulina flies. This shows that spirulina, by its antioxidant properties, helped in this fly model of Parkinson's. Paper 7, a spirulina-enhanced diet provides neuroprotection in alpha-synuclein model of Parkinson's disease, Pabon et al., 2012. A rat model of Parkinson's showed that a diet with spirulina reduced the number of brain cells damaged and the region of microglial, which are immune cells in the brain, in treated rats versus controls. These microglial or immune cells in the brain that can destroy foreign substances like viruses and diseased cells. So the spirulina rats had less of these cells seen, and there was less brain damage in the substantia nigra, which we talked about earlier. Paper 8, neuroprotective effect of spirulina fusiform and adamantine in the 6-OHDA-induced Parkinsonism in rats. Chattopadhyaya, sorry, probably didn't pronounce that right, 2015, Complements of Alternate Medicine. So amantadine is used to help Parkinson's disease patients only in conjunction with L-DOPA. These are drugs that work together. This rat model of Parkinson's showed that movement and motor activity was improved in rats given spirulina. So that was really cool. And also amantadine and spirulina were given together. Okay, so the next disease we're going to talk about is multiple sclerosis. It occurs in about 200,000 people in the United States per year. It causes people to have difficulties walking. Numbness may also occur in different parts of the body. Electric shocks can occur when moving your neck at certain times. Tremor and being unsteady also occurs. It's a neurodegenerative disease thought to be of autoimmune nature, meaning your own body attacks itself. It is a progressive disease that affects multiple areas of the central nervous system. It is a result of destruction of the critically important myelin sheath that surrounds nerve cells. So when you've got a neuron and you have a cell body and then it has ends to it and it's covered by this sheath called myelin. And that's important for the conduction of and health of these cells. So that's what gets destroyed, the myelin sheath. This sheath is mainly made of fat, hence the term white matter. It allows the nerves to conduct their electrical impulses. If they can't fire, there's no movement. In nerves for sensation, that would create numbness or electric shocks or dysesthesias or paresthesias, we call it. Its age at onset is between 20 and 40 years old. Females are 2 to 3 times more likely to have it than men. White races have a higher incidence in countries where temperate climates such as Canada, northern US, New Zealand, southeastern Australia, and Europe. A side note, I went to the medical school at the University of Western Ontario in London, Canada. At that time, a lot of research was being carried out in multiple sclerosis there because it had one of the highest incidence of multiple sclerosis in the world. Since I had a master's degree in immunology, I was asked to do summer research in the MS lab studying the cerebrospinal fluid in MS patients. We were trying to ascertain if there were different T cell populations and CSF of these patients. That was back in 1981. Still to this day, we're trying to figure out the pathophysiology of MS. Hopefully I contributed to something. Treatments include corticosteroids for acute attacks. Plasmapheresis or plasma exchange can be used if steroids don't help. There's an FDA-approved drug to modify progression called ocrelizumab to slow progression. And there's injectables. Interferon is an immune modulating disease. Glatiramer helps block the immune system's attack on the Myelin sheath, and oral meds, and there's numerous other things. Paper 9, ficosine and ficosyabilin as remyelination therapies for enhancing recovery in multiple sclerosis and ischemic stroke, a preclinical perspective by Penton-Wall, 2018 in Behavioral Science. There are cells called oligodendrocytes, or OG, which have precursor cells that go to the damaged neurons to help rebuild myelin. Rats given drugs to mimic MS received ficosine and had more of those oligodendrocyte cell activation and less myelin destruction than control groups. Rats that were given ficosine prophylactically, meaning preventively, did not develop the disease. So here we see neuroprotective effect of ficosine in disease prevention. Biopsies of nerve tissue showed thin abnormal myelin sheaths in the disease control group, but the ficosine group showed nice thick myelin sheaths. So what they looked at under the microscope, when they did a cross section, you could see that this was a nerve and that had a thin myelin sheath around it. And then the ones that were treated with ficosine had beautiful thick myelin sheaths. So it's really cool. They actually had some interesting biopsies. You can see these differences with electron microscopes. Biopsies showed that, and brain ischemic models to simulate a stroke also showed neuron improvement with ficosine. They also found an improvement due to reduction in inflammation because of the COX-2 inhibitor effect of ficosine. Again, synergistically working together. Last paper, promise. Fucoidan, a sulfated polysaccharide from brown algae, improves cognitive impairment induced by infusion of AB peptide in rats. Gao et al., 2012, Environmental Toxicology and Pharmacology. A rat model of brain injury showed that oral intake of fucoidan, which of course we know is in this, reduces oxidative stress and improved hippocampal damage in treated versus control. So I'm going to recap now. It's the end of my part in a minute, and then we have a special guest. So AFA, spirulina, ficosine, and fucoidan all improved the same way the damage to brain tissue. This was shown in multiple sclerosis models, Parkinson's models, and ischemic brain injury like stroke. SCU contains AFA, spirulina, and fucoidan. So in just one product, there are 3 potent repair ingredients for brain diseases. We saw the neuroprotective effects of myelin sheaths in Parkinson's cells improvement and ischemia. The oxidative stress reduction, enhanced immune system, stimulation of the oligodendrocytes, so cells that help protect, and the COX-2 inhibition all lead to improved brain health. To me, the thing that's so impressive is that SCU didn't just attack one problem at one level, it improved multiple problems at many levels. I think that's really critical here. So active, of course, contains spirulina, and so active joint contains spirulina plus boswellia. The latter is an anti-inflammatory. So again, more than one ingredient helping to improve brain disorders. So all in all, again, it's really unbelievable, as Andy calls it. I'm peeling back the onion, the layers of an onion, but why these products work. And there's a lot of, as you can see, great science behind this. So